Of Department of Plant Pathology, I welcome respect Director CPPS as well as the Professor Entomology, Dr. Satya sir. And I also welcome all the participants who are all worked in the field of plant production, who are all both in online and offline for oral presentation as well as the poster presentation. As rightly pointed by the Director CPPS and the Professor Entomology, it is a very good platform every year conducted by TNAU to train the PG students how you have to present the paper, how you have to write the paper, and get experience about that. And apart from that, I like to mention one important point. The topic itself indicates its own importance and significance. For example, food, farmers, and the future. So it covers all these three. So this is the nowadays all food, farmers, and future is the most important one. Why? Because the COVID-19 clearly indicated that the importance of agriculture throughout the world. This is known by all the persons who are living in the global region. Okay. So on behalf of Department of Plant Pathology, I wish all the PG and PhD students who are all going to present the oral paper and also the poster presentation to present in best way and get through all your presentation and win the awards also. Thank you.
Thank you, Dr. Muttamalan. In fact, we are late by 15 minutes. I'm sorry for that. The inauguration has been delayed because of that, this delay. Now I request Dr. Surendra Dara, Entomology and Biological Advisor, University of California. She is going to deliver a lecture on new integrated pest management paradigm for the modern age. Now it is over to you, Surendra.
Thank you. I'm trying to share my screen. 1 second.
Okay.
Okay, you can see it clearly.
Yeah, it is visible.
Okay, and I hope you can also hear me well.
Yeah.
Thank you very much, Dr. Kennedy and the rest of the team for inviting me to share this new IPM paradigm with you. And congratulations to all the graduating students. So we'll get started. If we look at the traditional IPM model, this is a lengthy definition from our university, UC IPM program. If you look at the definition of IPM, it basically talks about using all the cultural practices, biocontrol, host plant resistance, and everything possible before using something like chemical control. And we want to target only the pest species that is causing the damage, whether it is a weed or a pathogen or an arthropod. The pest is a general term, as all of you know. So here the objective is to control or suppress the pest using natural or alternative to chemicals first, and then move on to chemicals. As you see in this pyramid, you use everything and chemical control is the last option. But in reality, this is what you see. You treat the seeds, you treat the transplants, and you fumigate the field, and you do everything right from the beginning or even before the planting until the end with chemicals to suppress the pests. This is the reality. And if we look at the traditional IPM model, there has been just as we saw, the definition talks about maintaining this ecological balance in the system, in the cropping system, and applying pesticides or adopting control measures where it makes economic sense. So there is a balance of this ecology and economics as we see here. But in reality, but in theory, you see a heavy emphasis on the ecology. If you look at any kind of definition of IPM, you keep hearing that to do this to protect the environment and maintain the balance in the ecosystem and adopt control measures only when necessary, right? This is what you see in theory, a heavy emphasis on ecology. But in practice, you see a heavy emphasis on profits because a farmer has to make money. They have to be able to minimize their losses, increase their profits. So the emphasis there for them is economics. So there is this imbalance or disparity in what we preach and what farmers can practice. So it is a difficult thing. And traditional, if you also look at the traditional IPM, this is what we have, right? Like everybody knows these different components of IPM, the traditional IPM model where you have host plant resistance, planting resistant varieties, and doing all kinds of cultural practices to avoid pests or minimize or reduce the pest populations. And then biological control, either you conserve natural enemies or you can release natural enemies. Then behavioral control options are there using attractive materials or the baits or mating disruption. And then when it comes to physical or mechanical control, there are again a variety of options. You can use screens, you can use nets, or you can use certain kinds of traps, colored traps, or reflective mulch, or reflective materials, or vacuums, and so on. Then microbial control has been around for a while with, whether they're biofungicides or bio insecticides and so on. There are a variety of these materials based on microorganisms or their metabolites. Then finally, we have chemical control. Here, we usually talk about synthetic materials, but chemicals from plants or natural sources are also chemicals. So we have to put everything under this category, regardless of their origin. So as all of us are familiar, we have these various control options for suppressing pests. So if you are looking at the traditional model, you have this pest management and various control options as we discussed, but the heavy focus is on how much it costs and how much do I save. So because of this reason, although all these things, host plant, cultural, biological, and all this can be practiced throughout the cropping period, there has been a heavy emphasis on chemical pesticides because they're usually fast, they're usually effective, and they're cheaper. Because of this, the emphasis has been on synthetic materials. But are we covering everything? Just looking at this. Why do we need a new model? Because agriculture is not just about growing a crop for food purpose. It is an art, it is a science, and it is also a global enterprise. Whether it is a local market or international market, it is a business. So when we have an art, science, and business and all the social aspects, the affordability of the food, producing enough for everybody, and the safety for the environment and safety for human health and beneficial organisms, whether they're mammals or fish or beneficial arthropods and so on. We have to balance all these factors, human factors, social factors, and environmental health factors, business aspect and everything. Because of that, the old model no longer looks relevant. We're only talking about ecological balance and then just protecting the environment. That's what has been promoted, but in practice it is different. So we need to update ourselves just as we do that for everything. We needed to update the IPM model too. So this is what I published in 2019. So we have this IPM model where we have the pest management aspect, not just this one, the pest management aspect. There are 3 aspects, and the first one is pest management aspect. And within that, we have 4 components. One is pest management, then knowledge and resources, then planning and organization, and communication. Now we will look at all these 4 components individually. We already looked at host plant resistance and other control options, right? So we don't have to go over those again. But what about knowledge and resources? The pest biology is the most important thing when we choose a particular any of these. When we are choosing any of these control options, we need to understand what the pest is. Is it a plant fungus? Is it a bacterium? Or is it a virus? Or is it being vectored by an insect? And then if we are talking about an insect or an arthropod, where does it live? What does it infect or infest the roots or bore into stems, or does it defoliate, or does it skeletonize? Does it form leaf folds or rolls the leaves? So depending on those, and then whether it is whether it is the immature stage or adult that causes damage, and whether the adult has a sex pheromone or a mating disruption option we could exploit. We need to understand all these things. Based on that, we can choose one or more of the control options. So that is why we need to include that, the knowledge and resources, especially for the pest. Even in developed countries, we get asked questions that where we need to focus on this part, understanding the pest biology, so that we can choose different control options depending on the life stage or which is a vulnerable stage and so on. Then what kind of options are available to me as a farmer? Yes, there are all these options, right? But can I afford all of them? Or do I have a pesticide, or do I have a trap? Do I have an attractant or a bait or some kind of mating disruption option in my area or in my country or for this crop? Because in America and some other countries, not as you know, not all options are registered for every pest and every crop. You may apply a particular pesticide on a particular crop for a particular pest only, but not for the other ones. Even if it is registered for a crop, you may not use it against a non-target organism, even that organism is a pest. So there are those kinds of decisions you have to make based on the knowledge and resources you have, available options in your area. And finally, the tools and technologies. We cannot ignore the modern technology, whether it is modeling to understand when a particular pest is going to explode or when it can be effectively controlled with minimal effort, or using the degree days, or using sensors or drones to detect, or even using certain modern equipment to release natural enemies, or applying pesticides with the nanotechnology and so on. So we need to include those also as part of the IPM. Then coming to this planning and organization, monitoring is a very important thing. When there are large farms, there may not have been the farmer may not have enough resources to thoroughly monitor, do the scouting, all parts of the field. When you have smaller acreages like we have in India, it might be easier for the farmer to go around the field and check and see, determine whether it is time to, taken it up to spray pesticide or do something to control that pest. Whereas when we have large farms, like hundreds or thousands of acres, like in the US, it may not be easy to detect every area. So there might be a tendency to spray pesticides based on the time of the year. So now that 1 month after planting, we typically have this pest, so let us spray this. The pest population may not warrant that application, but we apply them based on the calendar or the seasonal, seasonal occurrence of that pest or disease, are based on our understanding of that. To avoid over application when there isn't enough pest, or applying all over the field when the infestation or infection is only in some areas, we need to monitor. When we monitor, we can apply that control measure where it is necessary. The next one is managing the information that we gather from year to year. When we have 10% of the field infested, maybe we use one of these options. And when it is 50%, maybe you need to use other ones or a combination of treatment options. And then these control options also, the efficacy of these control options also depends on various factors, the season and the environmental factors at that particular time and the irrigation condition and anything else, all kinds of biotic and abiotic factors. So we need to keep a record of all these things, when we did and how it worked and what was the most effective thing. So keeping records of this helps to build institutional knowledge to pass on that information, or even for us to use year after year. So that is why this information management is very important, especially when there is a larger farm. And finally, acting in a timely manner, whether it is sanitation or setting up some traps or applying pesticides, these need to be done in a timely manner. Otherwise, regardless of all these resources and everything we have, if we don't do the right thing at the right time, but pests may not be controlled as desired. Then the final component is communication, and here we need to focus on information exchange from researchers and other professionals to the farmers. So the farmers have to attend and also try to gather information from various resources to know about new pests. As it was mentioned earlier, there have been a lot of invasive pests all over the world. With the global trade and transportation and global travel, we have this pests spreading all over, so people need to be aware of this. And then also they need to know, people in the sense farmers need to be aware of what can be done and what is a potential threat. And then communication within the farming group, like if it is a small farm, it sounds very simple, but when you have a large farm and a lot of people working, communicating these issues, like all the information related to pests and their control, everybody should share that information within that farming company or that farming family. And exchange of this information and what strategy works well should also be communicated among farmers. Sometimes I'm not sure about the situation in India, but here, when farming is a big operation, sometimes the strategies they use are maintained as proprietary information, but pests don't have any boundaries. So it is very important for all the farmers to share this successful strategy so we can get area-wide control of a particular pest. And then most importantly, we need to maintain this dialogue with the public. Public in most cases is disconnected with food production system, what is involved and what is not only about the science, even the simple food production issues public doesn't understand. And that is very important because they are the consumers, they are the ones who influence how food is produced. So we need to take into account all these factors, technology and information management and communication, in addition to pest management. And at the core of IPM, we have research that we do either to develop basic information or fundamental information or applied information. We need to focus on both fundamental research and applied research, and then use that information or communicate that information to the growers through our extension or outreach system. So this is the first part, or the first aspect, pest management aspect of the new IPM model. Then we have the business aspect, where in the olden days, farmers produce and either they sell directly or they sell it to retail market and then the consumer buys. but it is changing. Like there are a lot of parties involved in between, and it is no longer the farmer. Here it is the producer who is deciding what to produce. For example, in America, it is the retailers, the sellers, or the retail market. They are the ones who decide how much and what and in what configuration and what kind of produce, which means either organic or conventional and all that. They are the ones who influence the producers, the farmers, to produce these items according to their business model. And then consumers actually don't have a lot of influence on what they buy. It is the retail market that heavily advertises on organic or some of these kinds of specialty foods because organic produce fetches more price. So they make more money. So they keep advertising about organic, and they tell these farmers to produce a certain percent of whether it is apples, strawberries, lettuce, or eggplant, or whatever they're producing, fruit or vegetable. They say we need 10 or 20% of organic. Only when you can supply that, we will also buy the conventional produce. So farmers produce according to the seller's demand and need, not influenced directly by consumer. Okay, and then how consumer is influenced is a big story. We'll talk about it later. So we have this influence business impact on this food production. Then finally, the environmental aspect. Here, it is not just environment. We have to maintain the environmental safety, human safety. In addition to that, we have to make sure that the farmer makes money. That is very important. A lot of we know in India, farmers have small land holdings, and they're not very rich like as they are in some other countries. So farmer has to have the farming as a good business so that they can make money, they can prosper, and they can continue to produce food that we all need. And at the same time, the social acceptability part is also very important. If you see, organic foods are expensive. Not everybody can afford, and at the same time, when we overuse synthetic materials, so a lot of people don't want to have them also. So we want to have a system where it uses all kinds of control options in a balanced way to get the desired control so that it is socially acceptable. Okay, so this is the new model published in 2019, and this is the version translated into Tamil thanks to Dr. Kavita Zedda. I have this model translated into multiple languages and a few Indian languages too. And I really appreciate her help in translating this to Tamil. And we published it in an outreach magazine last year. Okay, then with the new IPM model, what do we see? It is economically viable, it is environmentally sustainable, and socially acceptable. So these are the key points of this new model. And as I said, I published in 2019, and you can reach this article. If you haven't read, you can find this at this link, or using this QR code, you can take a look at that. And this article has been very popular, and it has been the most read article of Journal of Integrated Pest Management from Entomological Society of America. And I conducted a survey early last year where it is a kind of a worldwide survey about the impact of this new IPM model, and respondents indicated that they expect $34 million savings, and they expect to apply this information, the new model, on almost 1 million acres, and then communicate this information to this many thousands of people. So this model has been widely accepted and then used because it is not just about pest management. It also takes into account the business aspect and human aspect and social considerations and everything else. Now, I only showed certain things in that model, those two outer circles and then inner ones. But there are also certain factors that influence this model. Those elements are regulatory, commercial, and social. These are very important because no matter what we do, if there is a regulation for a certain thing, you all are aware that GMO crops are not allowed in India, right? Still not allowed. For example, the BT eggplant or BT brinjal is not allowed in India. Cotton, I think cotton is there, but some other crops are not. So how I will go over this quickly. So when we look at the regulatory elements, the pesticide registration status and how it is used is very important. That is, the government controls what kind of pesticide can be used for a crop and at what level, like when, where, how, and how much, how often they can use and how much they can use and so on. Then we also have these maximum residue levels, restricted entry interval, which means when after they spray, when they can enter back into the field, and what is the post-harvest interval, and all those things. At least here in the United States, those regulations help maintain the safety of the food. Wherever you have a system that works well, this kind of regulatory system, we can ensure food safety. And at the same time, advisor and applicator license here, not anybody can spray pesticides and not anybody can advise the farmer what to spray. They have to have a license, like a pharmacist or like a doctor, so they have to have the license. These are the regulatory elements, and then related corrective actions. If anybody violates those corrective actions, those things help maintain the safety, like the work behind. You don't see them in the circle, but this regulatory element works in the background. And then we have this commercial element, which is a very important thing. How much it costs to adopt all these measures, and the farmer has to have a good return on their investment. So that also that is not also, it is actually what mainly governs what kind of pest control option can be adopted. And then environmental cost. Not everybody pays attention to this. It could be cheaper for the farmer, but it might have a bigger negative impact on the environment. Or maybe it does not have that much of an environmental impact if it is used once or twice, but if it continuously used, we can have bigger negative impact. So these kinds of things need to be taken into account, as well as the producer, retailer, consumer part, the business part. Then the social element. Finally, we are towards the end of this, and the social element is the affordability for everyone. Most important thing. We have a lot of people who cannot afford nutritious food, but if we look at it, the cost of the conventional produce, it is not as much, right? Conventional is, we're not talking about whether everybody can afford or not here, just looking at it. This is typically conventionally produced fruits and vegetables have X amount, but organic produce is always more expensive. But not everybody can afford this. So if we have a system where an IPM based system, which could cost about the same or maybe slightly higher. Here I put about the same because if you use chemical or synthetic materials continuously, they become ineffective with pests developing resistance. So in the long run, it could cost more. So when you follow IPM principles, then it is a balanced approach and it optimizes the cost. So this is better and it can ensure affordability for everyone. Then the next part is food wastage. It is a lot of food is wasted due to logistics, marketing, or consumer preference. And now in India, it is because of the politics in some states, people produce and then they throw all the fruits or vegetables because of the market conditions. It should not happen. But there is plenty of food wastage here. This is they both are in California. Here you see all the citrus fruit is not marketed because because of multiple reasons. It could be the price has fallen down, or maybe it costs more to ship them to the stores and so on. So all of this is wasted. And here it is the cabbage. Some of them are damaged because of a disease, and then some of them are healthy, but they cannot just go and search for it. It takes too much time for the workers to look for those. So they abandon the entire field. And similarly, the consumer preference for certain weight and shape and appearance and all this also is putting a lot of burden on the farmers, because it is not like a manufactured item. It is a grown fruit or vegetable. You can't expect them to have uniform size, but you have to market them in a particular box with certain numbers. So because of those reasons, there will be some wastage. And if you look at these, the strawberries on the left, these are ripe and taste good, but they're not marketable because of this deformity. That is how the consumer preference is. And here these cherries, it is very common to have double cherries, or sometimes because of environmental or genetic factors or other reasons, and also have these projections. And these are not marketable. This slightly in this picture, it looks different, but this one is a double. It is just like a double cherry. So, but it doesn't fetch as much money. Because of that, these strawberries are thrown usually. These are sold for a lesser price. And here these eggplants or brinjal, they look fine. We can cook them without any problem, but usually they don't fetch as much price because of that projection. So we have these kinds of issues. And then another social element is the public does not know, as I mentioned, they don't have enough knowledge about food production, what is organic, what is conventional, what is natural, and all these systems they don't know. There is a big problem of chemophobia. You can be consuming all kinds of chemicals, taking all kinds of medicines, or drinking alcohol and doing everything. But when it comes to fruits and vegetables, everybody is afraid of pesticides. So, and because there is a lot of false information, so there is also the reason we had that public education or public outreach is for this, so that they can be educated and make the right choice. So if you look at if you Google organic, or if you Google GMO and look for the images, this is what you see. Organic is good, they promote, and they say pesticides don't know when to stop killing, or they're poisonous. And then GMO means they're being injected with something very dangerous. And all these kinds of things make people worry about things that are necessary. Like all this is kind of false information, not science-based. It is fear-mongering, and that puts a lot of burden on the farmers to produce. And if we don't follow IPM, then pest problems can become area-wide issues, and food costs increases, productivity reduces, and so on. Because of all this, we need a good system, and that is IPM. So if we have, instead of having an organic label, organic produce, if we have an IPM based label, it is safe, it is profitable, and it is practical. And that gives everybody the confidence and ensures profitability for the farmer and safety for the consumers. And if you have time, please take a quick minute. You can take a photo of this link and then give your feedback if you think this IPM information is useful for you. I appreciate that. And with this, I end my talk. I think I have 10 minutes or so if anybody has questions.
Okay, thank you, Dr. Surendra, for nicely presenting the new IPM strategies to be followed. In fact, even the old IPM strategy or new IPM strategy, whatever is there, the growers, they have to decide what kind of a thing they have to go for. As you said, the social element, then environmental safety and all. We are all talking about all those things for the past so many years. In spite of that, the growers, as far as India is concerned, the system is totally different when compared to the US.
That's
When
right.
you know, if you ask them to go for this kind of spray, this kind of IPM and all, that can be followed in US, but in Indian system, it is not being followed fully. Even though the IPM strategy we have introduced long
Has been
before,
there.
in
Yes.
spite of that, the growers are not following it. Very few farmers they are following it, and most of them they are using only the insecticides or pesticides or fungicides or whatever it may be, chemicals.
That's right.
So now people in our state, especially in Tamil Nadu, people have started talking about this organic farming, and even for some of the pests introduced, invasive pests, we are requesting the farmers not to spray insecticides, but in spite of our request, the farmers are doing it because, for example, in the case of rugospiralling whitefly in coconut, we are asking them not to go for any spray, insecticide spray, which will affect the predators and parasitoids present in the ecosystem. In spite of that, many of the farmers, they are just doing it.
That's
So
right.
we have to create, we have to change the mindset of the farmers. So that is the only option. Yeah, thank you for nice presentation, and hopefully we will follow the new IPM model which you have presented in future in Tamil Nadu and in India also. Thank you very much, Dr.
Thank you.
Surendra,
Thank you very
for
much.
joining with us. Next, I request Dr. Mamta Sharma. Connect பண்ணுங்க. Dr. Mamta Sharma.
Hello.
Dr. Mamta Sharma.
Yeah, I'm here.
Yeah. Good morning. After a long time, we are meeting again. You
Yes.
gave a guest lecture or something like that to our students. So on that occasion, we met. Now I request Dr. Kennedy to introduce Dr. Mamta Sharma.
Good morning, participants. Good morning, Mamta. It's nice to see you over here. Dr. Mamta is a principal scientist at ICRISAT. She's basically a plant pathologist, and also she is now into climatology, and she has got a major project in climatology. And right now, TNAU is also collaborating with ICRISAT with Mamta's project, and we are working on that in developing models for pests and disease occurrence, phenology-based pests and disease occurrence. Now, Mamta would be talking about.
She is going to present on genome analysis of pathogenic microbes. We are really interested to hear from Dr. Mamta about this particular topic, because this is one of the important topic. In fact, most of our pathology students, and as well as even the entomology students who are all working on the biocontrol or something like that on microbes, I think they'll be benefited by your lecture. Now I request you to present it, Dr. Mamta.
Thank you so much for giving this opportunity to give this presentation for the students. And although, as Dr. Kennedy mentioned, I work on the climate change and other areas, but I will be talking right now in a different domain on the this genomic analysis and how the genomic tools are used in case of the microorganisms. So I'll share my presentation. Hope this is visible.
Yes.
Yeah, it is visible. You just make it full screen.
Yeah, I'll do that.
Yeah, now it is okay.
Yeah, so just directly going into the talk. So I'll be talking about that. What are the different genomic tools and how they help in the plant pathology, and giving some insights on whatever work we have done at ICRISAT. Maybe that will be beneficial for the students in conducting their research in the different doctoral or the master's program. So first thing comes in mind that what are the different areas that I will be talking about in this talk. I will be dealing with how the genomic tools that help in the detection of the various pathogens, and then what kind of the evolutionary relationship or the understanding we get from this kind of the tools that help us in understanding that where we are closely related with our ancestors in terms of the microorganisms. And then also, I will be dealing with the kind of the epidemiology and how the tool help us in quantification of the different pathogens when it invade the host. And some of the examples, what we have done with respect to the host and the pathogen, with respect to the use of or the application of these genomic tools. So my talk will be revolved around these topics, and towards the end, I'll be happy to have some discussion around these areas. Moving on to just to give an idea, what are the different tools? And in case of when we say about the genomic tools or the nucleic acid based technologies that are at presently being used in this domain. So we know all about the different DNA markers that are already being used for the detection of various pathogens. And then some of the PCR based approaches or the techniques that are used. Then we have a next generation sequencing based approaches like whole genome sequence and the transcriptomics, proteomics, etc. These are the another technologies that are being used for understanding the various host pathogen interactions or the pathogen itself. And then some of the more advanced tools, for example, where we not only need this PCR based approaches, but then based on very rapid and quick methods that can help us in the detection of the pathogens. And as I mentioned, the quantification of the pathogens based on the latest approaches of the qPCR and on. If I talk about first thing, like how these tools helped us in detection of the various pathogens and diseases, I have taken here an example of the chickpea. For example, we know so far we detect the pathogens based on the visual detection of the symptoms, and then we go into the mycological identification of the pathogens. But what happens, what are the disadvantages of these things is that if the several diseases or the complex diseases are happening at the same time, sometimes it do not give us the accurate pathogen identification, and we end up identifying in a wrong way, and then a whole management strategies is disturbed. So how these molecular based tools that help us in quantification or the accurate identification, that is the biggest advantages what we have been offered by these genomic tools. I'll just given how the technology has advanced over the period of decades. In past, I think in 2000, early 2000s, or till now also, we are going into the mycological detection. We use the microscope for the morphological basis and the cultural identification of the pathogens. But then we moved into the era of the PCR based or the marker based identification of the pathogens. And then further, we moved into the direction of the just visually, if we have the DNA, very small amount of the DNA of the pathogen, we can quickly identify within a like less than an hour the what is the causal organism of that particular disease. So that's how the technology has advanced over the period of years in this area of the plant pathology. Then I'll just cite few examples here where we have done the identification using this genomic tools, and I will take you through the different techniques that we have used in this. For example, the first one is the PCR based technique, where we use this technology, particularly when the two pathogens, for example, in chickpea, we have two, in pigeon pea, we have two pathogens, Macrophomina and Leishmaniasis. Both they look similar. Both the morphological identification of the pathogen is very, very similar, and it becomes really challenging to accurately identify that what is the exact causal organism. There, the specific PCR based approaches or the primer based approach that has helped us in accurately differentiating both these pathogens and giving a high level of the accuracy for the identification, even based on the size of this thing. If you say 220 base pair for Macrophomina fasulina and then for Leishmaniasis 178 base pair, this can help us in quickly deciphering that the pathogens. And in fact, we got the distinct clustering for both the pathogens and could identify the exact pathogen which is causing the disease. Then another approach is the ITS-based sequencing approach, which is the most commonly used approach nowadays and it is now essential for in every manuscript what we publish that we should have a molecular identification of the pathogen in that reported. Otherwise, the acceptance of that paper is rejected and they ask for the molecular identification. So this is the approach which is most commonly used nowadays for the identification of confirming the particular identification of the pathogen. Again, I cite an example of the Phytophthora blight disease, which is caused by Phytophthora cachini. Here, a little bit history about this, just to give you an idea that how accurately we get an information based on the genomic tools here. So there are two species associated with the disease of this Phytophthora blight. One is Phytophthora dreschleri, another one is Phytophthora cachini. If we identify them morphologically, you will see we'll not be able to differentiate that whether we are talking about dreschleri or whether we are talking about cachini. However, based on the specific ITS primer that has been designed for this pathogen, we could clearly disintegrate that whatever the isolates present are present in our major pigeon pea growings in India, all those isolates that resemble with the Phytophthora cachini, and not even a single association was found with the Phytophthora dreschleri. And that is a kind of a very specific information we got on ascertaining the role of the pathogen in causing the disease. So this is the paper what we published based on this, where we reported that now whatever the isolates that are present in India, that is in case of pigeon pea, is Phytophthora cachini and should not be confused with the Phytophthora dreschleri. Another approach, what I said in my initial talk, was the lamp-based technique. This is a visual technique and very rapid one, and a lot of studies are being conducted now in this way. So there are several publications that has come up recently in this area where they have researchers have used this lamp-based, which is a loop-mediated isothermal amplification method. It is like the advantage of this method is that you can visually see that the presence of the pathogen, and it is very accurate and specific to the host or the pathogen. Very, very accurate, timely, sensitive, and even very low amount of the DNA, we can quickly diagnose the pathogen. So that's the advantage of this lamp-based tools. And here I have given two examples again. Fusarium species in case of chickpea, and again the Macrophomina faeculina in case of chickpea, where you can see that the color changes that depicts the presence of the pathogen. Where there is no change in the color, that is the absence of the particular pathogen in that infected sample. So in that, if you see within 75 minutes of the timings, we can detect the presence of the pathogen from the infected plant sample as well as infected soil sample. So that's the advantage of all these kind of latest technologies, and further improvement are being done in all these approaches to still minimize the timings and lead towards the biomarkers based approach, that those kits can be used in, for example, I see in next 5-6 years, all the KVKs and all those will be equipped with these kind of the kits in hand and can quickly identify the pathogens present in the field itself, that that will give a real-time detection of the pathogens in a very minimum time. Then another one step ahead, the technology has gone, where now we can see inside the host also how much pathogen load is present in the particular plant or the particular variety or the crop which is being infected by the pathogen. So that is the quantification based technologies that has helped us not only in the detection, but also in quantifying the colonization of the pathogen inside that host. Again, we did one study where we were studying the impact of the, for example, elevated carbon dioxide or elevated temperatures on the how it's infect the pathogen load inside the host. So we took two different genotypes here, the resistant and the susceptible one, and trying to see the pathogen load inside both susceptible and the resistant one. So what we could see here is that in case of the susceptible one, in case of Fusarium wilton chickpea, at 7 day, even when the symptoms were not present, we could see that the pathogen has started invading and there was some amount of the load already present in the host plant. And at 14 days, it completely expressed with a high load of the pathogen inside the plant. However, in case of the resistant one, for example, here the resistant variety is WR315. Although the symptoms are not expressed, but the pathogen is present to a minimum extent inside the plant, which again gives us an indication about the defense related genes that are present in the resistant plants as compared to the susceptible plants, which are helping us in counteracting that effect. So what I'm trying to say here is that these approaches like qPCR based approach that gives us an exact indication that to which level in the plant the fungus has reached and how much is the load of that fungus inside the plant, which gives us several leads towards understanding the host pathogen interactions inside the plant that is happening. So I just given another example where we are trying to see like in the era of the climate change, a lot of changes are happening, and then soil moisture is the another important that plays a major role in case of the increasing in the soil-borne pathogens at present, which were not to be seen earlier. So we did one study to establish that the cholera rot is the one fungus which is it's like it infects wide range of the host plants and is becoming a major problem in several pulses as well as I see in a lot of vegetable crops also. So a study was done where we could one thing detect we using the ITS based primers that easily the pathogen, and the another one is we could see that how much is the inoculum load inside that plant of the pathogen. For example, here we have done one study where when the moisture is high in the field, the disease outbreak is more. And we could correlate it the pathogen load also inside the plant, that there was a high load of the pathogen inside the plant. So severity can be directly linked with the colonization of that plant with these pathogens. Now, another area where the genomic tools that play a major role is the understanding the pathogen variability. I think a lot of studies by most of the students are done is that what is the diversity of the different pathogens that is present in case of different crops. So the genomic tools that play a major role in understanding the diversity or the different races or the variants or the pathotypes that are existing in the different crops. So I'll take you through some of the examples in that direction. All of you know that when we grow the different varieties in different environment and in different areas, so there is a variability that exists in that isolates aggressiveness or the virulence pattern. So in that direction, how to understand that whether whatever the isolate or the of that pathogen is present at one location, whether it is same at the other location or is it different. So that kind of investigations so far is being done using this molecular variability, cultural variability, and then pathological variability. A lot of studies we do based on this and see and try to identify and report that in that location, the isolate or the race present is similar to the one which is present at the different location. So far this study is being done using the cultural, morphological, and even the host differentials that are used to decipher these races. But there is a lot of movement in that direction, and I'll just quote here a few examples here where a lot of markers are being used to understand this molecular variability. And this has been started right from the AFLP, moving on to the RAPD, SSR, and ISSR, etc. These markers could differentiate between the pathogenic and non-pathogenic isolates, and also give some indication towards the grouping of the isolates with respect to the location. But still, there were certain disadvantages that we were not able to get the location-specific grouping which matches with that particular race. So again, there is some advancement that is being made in this area, and there are now these platforms available which could clearly differentiate between these race-specific markers or the variant-specific markers that helps us in devising the location-specific disease management approaches or facilitate the breeding for that location. So one such technology is also this DART platform, which is called a diversity array technology. And this technology was developed by Australian group and was initially used for the various genotypes in the crop improvement program to see the clustering of the genotypes with respect to the location and all. So we did first time use this technology for the pathogen and again for the Fusarium oxysporum from a species size series in case of chickpea. And we developed this DART arrays and try to profile the around 61 isolates and from all over the major chickpea growing regions and see what is the polymorphism or the diversity present in that. We could clearly get a differentiation, as you can see here, four different groups that are present in all over India in case of Fusarium oxysporum species for case of chickpea. But also this gives us an indication, if you see in this slide, one blue color complete band, that is that race 1 isolates. So we get an information that race 1 is the predominant race, which is present in all the chickpea growing regions in India, and followed by this red color band here. You can see these are all isolates that has been grouped together by using this different DART markers. So we not only get the information about the predominance of the race, but could get clear information that these are the different clusters, distinct clusters, which represent the particular race. Now, what is needed further is that we know the specific race-specific alleles or the markers, but then there is a need to move further into this direction and convert them into the biomarkers and use them like regularly in our programs to quickly identify these race specificity of the different pathogens. Now, another approach is that this technology is moving very fast, and then next generation sequencing approaches are being used for understanding the resistance and the susceptibility or the host pathogen and environment interactions kind of studies. So one such approaches are the whole genome sequencing and whole transcriptome analysis that is being recently or very widely used, and a lot has to be done in that area to get more information about the role of the particular candidate genes in with respect to the virulence or the defense relationship. So I'll just give you a few examples here. This is the whole genome sequence of one of the pathogen, again in case of from India, that has been done for Fusarium oxysporum, again in chickpea, with the group from the Australia. And then comparison between the three species was done with the Medicago and pea and chickpea pathogens. And it was widely compared with the tomato species. So what could we see clearly from this whole genomic sequence information is that all the legume related species like Medicago, pea and chickpea, that has a greater sequence conservation as compared to the Lycopersicea or the tomato infecting pathogen of the Fusarium. Another information what we got from this was that there are some specific genes like secreted in xylem genes which are only present in case of the chickpea infecting that, not the pea and Medicago. So that is a like getting a clear information that there are some specific virulent related genes that are causing the pathogenicity in case of chickpea, which is not present in the other species. So that's the kind of information we got from this whole genome sequence of the pathogen, and I think it is very useful to further move into this direction of understanding those genes, their role in the pathogenicity. So I'll little bit go deeper in this in one or two slides that how these secreted in xylem genes, what we got from this whole genome sequence information are helpful in this pathogenicity role in case of the different. So if you see here, these are the genes, as I mentioned, prevalent in case of the chickpea. And when we try to understand the presence and absence in all the isolates that are present in India, we could see that in some locations, some of the isolates, only few genes are expressing, and in some of the isolates, only few genes are expressing. So that again gives us an indication that the isolates that are present in, maybe I'll just in the south of India, are having different genes that are responsible for causing the pathogenicity as compared to the isolates that are collected from the central India. So again, it takes us towards the race specific or the variability that is present in the different isolates. And we could even identify the specific genes that are responsible for that. Another example is that how to see this their expression. So I just, if you see here the graphs, that we here did the expression of the 4-6 genes, the 6-5, 6-8, 6-11 and 6-13 in the susceptible and resistant genotypes. And what we could see here is that the regulation or the upregulation was more in the early stages of infection as compared to the later stages of infection. And even we can see that if you see here the 6-8 genes, that was 1,000 times upregulated. So which gives us a some leads towards developing into the biomarker using this particular gene that will be able to help us in identification of some race specific isolates or the race specific pathogen variability. Further, I think we did one again a little more studies in this direction to see the relationship, how much it is closely related with the other legume species. And again, we found that this particular gene was closely related with the model, I think the pathogen in case of the Medicago. So not the other pathogens of the Lycopersicea and other Fusarium species. One more example I would like to give the another technology, the transcriptome, which is also now being used by several students or the researchers to identify some of the candidate genes which are responsible for the resistance and the susceptibility also. This is another study that has been done with respect to the microorganism. And here, because we all know that conidias are responsible for causing the disease in case of some of the pathogens. So what is that triggers inside that conidiation process that helps in the infection process, or this whole process takes us to understand that what is the kind of the different genes that play a role in the early stages of infection, and then what are those that helps us in the invading that host to cause the disease. So all these kind of information we get from all this transcriptomic analysis of the different pathogens at different time points. And the interesting information what we got from this study is that there is a rapid shift in the gene expression when this conidia invade the pathogens. Another interesting thing what we got was that there are different pathways that are being triggered, or the changes happen in the different pathways, which yeah. Then there are different pathways. Can you see my slides now? Because I see some problem.
Yeah, it is there.
It is there. Okay. Because I think something happened and yeah. So, and another interesting information what we are getting is then these genes are transcribed in some successive ways. So at early time points, the genes which are responsible for the sporulation that get activated. However, at later time point, the genes which are responsible for invading the pathogen that get activated. So all this information is again helpful in understanding the host pathogen and environment interaction processes, and also that gene for gene hypothesis that is also understood at the molecular level using all these different approaches. So another example wherein we can see now both the corresponding virulence and the defense related genes, we see very clearly that if we see, I'm giving an example of again Sclerotium rolfesi that infect the chickpea, and then what kind of the variation that that occurs in case of the expression of the in case of the pathogen and in case of the host, the changes that is happening. So we see in the virulence related genes like lactin that is highly upregulated in case of Sclerotium rolfesi even after the 12 hours of the infection. And at the same time, the defense related genes, for example, here lipoxygenase that is highly regulated during the course of infection. So again, the gene for gene hypothesis that play a major role, and that is very clearly we can understand those entire processes using these recent approaches of the transcriptomics and whole genome sequencing and all. So with this, I mean, I just was giving an information about the different application of the tools in the detection, as well as in understanding the variability of the pathogens, and then leading towards the whole genome sequence transcriptomic, little bit going into the deeper to understand the infection processes and what is happening inside the host plant and in the pathogen. So just if I summarize all this, that what is the future of whole genome, this genomic tools in, so I see their major application in the detection of the pathogens. So that's where that can play a revolutionized use, wherein the pathogen can be detected within a very minimum time period. Otherwise, if you go through the standard conventional approaches, it takes several days. We need to bring the pathogen to the lab, isolate it, and then culture it, and then identify it. So that's a time consuming process. However, if we can rapidly identify the pathogens even before the symptoms appear in the field, that is going to be the revolutionizing thing. And I see a lot of opportunity in this direction, which will help us in the timely management of the diseases, and I see insect pests also to reduce the losses caused by them. So with this presentation, which I try to simplify in so that it becomes easy for the students to understand, I will close here and would like to thank you again for giving this opportunity, and happy to take any questions or the discussions. Thank you so much.
Thank you, Dr. Mamta Sharma, for explaining about this, genome analysis of pathogenic microbes. In fact, previously, based on the morphology only, we used to say this is based on the spore structures and other things, we used to say that this is the pathogen or something like that. Even if some strains are there, we could not able to differentiate what actually it is, whether it is a strain of FOC. Now, because of this advanced biotechnological tools, now we can identify which strain actually, what difference is there in their genetic makeup and all we are doing it as you have summarized. So this is a very important tool. Now, one of our scientists, Dr. Kamalakannan, is working on the LAMP assay for the reduction of this, grape powdery mildew and downy mildew. And he is also using some other management or direction. Rice airborne pathogens also he is detecting using this LAMP assay. As you said, even before the production or even the presence of symptoms in the field, we can, with the available DNA or RNA, whatever it may be, with that we can identify the pathogen, which we can very well forecast to the farmers that you have to take up this management aspects, or otherwise your crop will be affected by this particular pathogen and all. So this is a highly useful tool by which we can help the farmers by forewarning them that your crop may be affected by such and such disease, and you have to take up the management aspect. I thank Dr. Mamta Sharma for sparing his valuable time and presenting a valuable topic, and it is an eye-opener for our students also. In fact, most of our students are participating in this particular conference, so it will be definitely it will be a helpful one for them to proceed in their research program. Thank you, Dr. Mamta, for spending time with us.
Thank you so much. Thanks a lot for giving this opportunity. Thank you. I can stop sharing.
Thank you. Yes.
I can leave the meeting, right?
Yeah, you can.
Okay. Thank you so much.
Thank you. Now just we'll move on to the oral presentations. First presentation is by Mr. Ayyadurai. Ayyandurai.
Sir, my voice is audible, sir?
Yeah, it is.
Yes, sir.
Yeah, student.
Sir, my screen is visible, sir. Shall I present, sir?
So you have to present for 10 minutes and 5 minutes for discussion. Okay?
Yes, sir. Good morning to another present here. Now I am going to present a GCMS analysis of antimicrobial compounds present in the Mahu oil cake against the stem rot pathogen, Sclerotium palsy. Granite is a king of oilseeds. It production 7.3 million hectares, and the production is 6.72 million tons. So our India is leading producer after the China. China is the 70%. Our India is 52%. So the stem rot groundnut is suffered with more than 55 pathogens. So among them, the stem rot of Sclerotium palsy cause the 10 to 40% yield losses per year. So these pathogens to make the symptom is yellowing and wilting of the plants and rotting of the stems. So the white color mycelial growth, cottony growth forming on the stem region of the plant, and then in case of the pig formation and pod formation stage, everything will be lost to the yield. So nowadays, in case of the chemical management, the chemicals residues present in the soil, and also the pathogen may cross resistant to the fungicides. So the current scenario of agriculture, we are to moving on to the biological management, like as a plant products and eco-friendly management, like as organic amendments. So in this condition, the oil cake has a potential role to overcome that particular pathogens. So the oil cakes have a some fungi toxic molecules and and another way some volatile molecules are produced, volatile compounds are produced, and enhance the antagonistic fungi present the soil means that also enhanced by the that oil cakes, and also their nutrient soil fertility also improved and provide the essential nutrients to the plants. So these are way they are acting in the soil. So in this research, we have to frame the objective survey and isolation of the stem rot pathogen and proving the pathogenicity and molecular confirmation and assessing the efficacy of different oil cakes like as a castor, mahua, neem, and other oil cakes extract against the sclerosing policy under in vitro condition through poison food technique. And the best performed oil cake extract in the poison food technique, it powered to the Gcms analysis for any fungi toxic biomolecule compounds present in there. So the survey of pathogen, the pathogen is isolated from 5 different districts of 10 different places, like as Sivagangai, Madurai, Theni, and Tiruvannamalai, Salem districts. So among them, the block of Theni district have the high disease incidence. So in that particular place, the symptoms occur in from the 10 to 15 days after sowing plants, and also the fake formation stage. During the fake formation stage, the second image, the fake formation stage, everything will be damaged. And the third image, so after the harvest, that produced to used for the feed for the animal, that also entirely colonized by that particular pathogen, and for the field is fully infected by that particular pathogens. So this image is a pictorial representation of the work what I doing in that particular research. So the isolation and molecular confirmation of the pathogen. So that pathogen already the virulence is confirmed through pathogenicity. Then particular condition, the 89.27% disease incidence that produced in the fat culture conditions, and also in case of the petrified within 2.5 days they are covered the entire petrified and producing the more than 400 clerosia per plate. So that pathogen are molecularly confirmed that DNA is extracted and sequence is sent for sequencing, and that sequence is deposited in NCBI database and got the accession number. That particular virulent culture is virulent culture used for the analyzing of oil cake extracts. So that oil cake extracts are best assessed against the sclerosing policy. So the best performed oil cakes, the secondary fungi toxic molecules are analyzed through Gcms analysis. That here using the solvent is hexane solvent used for that to extract the secondary metabolite present in the mahu oil cake extract. So these are the poison food technique, the various oil cakes, pungam cake, neem cake, coconut cake, and mahua cake and castor cake. Both 5% and 10% concentration, we have assessed the metabolites against the sclerosing policy. The 5% and 10% concentration to effectively to reduce the mycelial growth of sclerosing policy, in followed by neem cake. So in this table, to each and every cake to vary the reduce the mycel growth of the sclerosing policy. The mahua cake reduce the mycel growth 1.5. They produce some mycel growth 1.57 centimeter. In case of 10%, they produce some sclerosing policy mycel growth is 1.29 centimeter. That the reduction percentage is 83.33% in 5% concentration, in 86.66% in 10% concentration as compared to the control is followed by the mahua cake, followed by the best performing oil cake is neem oil cake. The best performed oil cake, mahua oil cake, the secondary metabolites are analyzed through Gcms. The hexane extraction is followed for the Gcms analysis. So in this condition, the 9,12 octa decadenic acid, hexadecanoic acid, methyl ester, and octadecanoic acid, 12 ester. So these are the major compounds, high peak area percentage and retention time. So these are the compounds are to effectively to reduce the sclerosing policy and other specific role act as a antifungal activity, antimicrobial activity, and nematicidal activity. The many authors are reported. So against the sclerosing policy. So these compounds are further, we have to extract it through column chromatography, like as a TLC and NMR. So in this basis, these compounds are extracted, and that compounds are based formulation we have to use for to control the sclerosis pathogens. So result and discussion. In this condition, the Mahu oil cake extract produces the minimum mycelial growth 1.57 centimeter, 1.29 centimeter in with maximum percent growth reduction is 83.33%, 86.66% at 5% and 10% concentration respectively. So the particular compounds like I say hexadecanoic acid, octadecanoic acid, this also evaluated through the GCMS analysis. So apart from these compounds, more than 16 compounds are analyzed through the GCMS, squalene, so methyl stearate and other phenol compounds, and these are also evaluated through the Gcms analysis. So these compounds having Mahu oil cake combined with our Trichoderma species, that will be forwarded to our pot culture and field conditions. So at the same time that the octadecanoic acid, hexadecanoic acid, these major RT value and peak area percentage containing compounds are separated through the column chromatography and NMR based. These compounds will be based formulations will be overcome for the disease. In future research will carry on. Thank you, sir.
Thank you, Dr. Ayendurai, Mr. Ayendurai. Now the floor is open for discussion. Any questions? Go to the slide in which you have tested the 10% and 5% concentration, you know?
Yes, sir.
Mr. Ayendurai, that
Yes,
slide
sir.
you
Yes,
put
sir.
it. Next one, the table.
Yes.
So other oil cakes are also equally good when
Yes,
compared
sir.
to Magua and you know, this only
Sir,
poison food technique.
yes, sir. Poison food technique.
How you did it? How you did this experiment?
Sir, the Mahu oil cake, 1 gram of Mahu oil cake with sterile distilled water, 1.25 ml ratios will taken, sir.
Mm-hmm.
So likewise, we have take the 5% concentration and 10% concentration.
So other things are also oil cakes, pungam cake or neem cake or something like that, or they're oil?
Sir, oil cakes only, sir.
Oil cake only.
Yes, sir. Oil
Okay.
cakes extract only, sir.
So this Mahua oil cake is available in plenty.
But
Where
yes, sir,
did you get
nowadays,
it?
sir, from the local market of the Madurai, sir.
Is it available in?
Yes, sir.
Huh?
Yes, sir. Yes, sir. Available,
Are
sir.
you sure?
Yes, sir. Sure, sir.
Once we come out with the recommendation of all these things, we have to tell the farmers where they can get it. Even neem cake, we are facing a lot of problems. Neem cake is available in plenty. That's what we were thinking. But when we when we wanted to purchase it, we faced a lot of problems with the neem cake. I don't know about Mahua. Mahua cake, if we go for recommendation of this Mahua cake, then I think it is a little bit difficult. Dr. Muthamalan.
Ayendurai?
Yes, sir. Yes, sir.
Yes. So you screened almost 5 organic product,
Yes,
and out
sir.
of
Yes,
that
sir.
you selected the Mahua cake based on your in vitro
Yes, sir.
experiment.
Yes, sir. Yes,
And
sir.
finally, you identified what are all the compounds present by using GCMS and other thing.
Yes, sir.
My question
Sir,
is,
yes, sir.
yes,
Yes, sir.
yes. So here you used the latest techniques which are available in your lab or in the ACN or Madurai. It is very good, but
Yes,
my
sir.
question is, Have you compared the effect of antifungal compounds you have identified from the Mahua cake with the antifungal compounds identified from the biocontrol agents? Among these two, which one is most effective? Or how much quantity of antifungal compounds you can get more from either from a biocontrol agent or from your organic product? Do you have any idea on that?
Sir,
Or based on the literature you have studied so far in your research work? So why? Because as rightly asked by Director CPPS, of course your Mahua oil cake may play most effectively. But if you
Yes,
come
sir.
to a recommendation, if it is not available in large scale in the market, we cannot recommend your Mahua cake for a management aspect. That's why we are asking.
Yes, sir. Yes, sir.
Whether you have compared or not with the biocontrol agents. Or have you any information by read the material already published in the published articles from many journals?
Yes, sir. Many articles published, sir. But the compounds will be detected like as octadecanoic acid, 9,12 octadienoic acid.
No. My
These
question
compounds,
is, you have identified the antifungal compounds present in the Mahua oil cake. Okay.
yes, sir. Yes, sir.
Similarly,
Yes,
the
sir.
antifungal compounds already identified from the biocontrol agents also, for example, trichoderma, pseudomonas, anything.
Yes, sir.
My
Yes, sir.
question
Yes, sir.
is, among the antifungal compounds identified or isolated from your organic product, that is Mahua oil cake, as well as from the biocontrol agents, either from trichoderma or pseudomonas or any bacillus, anything, among
sir.
these, among the antifungal compounds identified from the oil cake from the biocontrol agent, which one is most effective?
Sir, oil cakes, sir. But here the peak area percentage is very high, sir, in compared to the trichoderma. From the I'm here searching some articles, sir. Here the peak area percentage of that octadecanoic acid and other compounds, sir, the peak area percentage is very low, sir, in compared to Mahu oil cake, sir.
No. Because that's why it is not so. But the compounds identified from the biocontrol agents are most effective. That's why it played a more vital role when compared to the organic products. Please kindly go through the article. Thank you. Thank you.
Yes, sir. Yes, sir. Thank you, sir.
Ayan Bhai.
Sir, yes, sir.
So I have I want a clarification.
Yes, sir.
So the primary source of inoculum for this Sclerotium ralpsi is this Sclerotia, which is present
Yes,
in the
sir.
soil. Have you
Yes,
tested
sir.
the effect of Magua oil cake against the germination of this Sclerotia? Because
Yes,
that
sir.
is the,
This also.
have you tested the against
Yes, sir.
germination of Sclerotia?
Yes, sir. I tested, sir.
Okay.
That mango oil cake extract also to inhibit the germination of the sclerosis also, sir.
Okay. Then one more clarification I need. So this Magua cake, when applied to the we are going to apply to the soil only. Okay.
Yes, sir. Yes, sir.
So when this Magua cake effectively inhibit the Sclerotium ralpsi, what will be the fate of our Trichoderma viride or any other beneficial microorganism present in the soil?
Sir,
So
it
that will be
enhances the trichoderma.
Have you tested the effect of Magua cake on the beneficial microorganisms like Trichoderma viride?
Yes, sir. Yes, sir. That pot culture study I have to carry it out, sir. That enhance the antagonistic activity of trichoderma, sir. Trichoderma population also they are enhanced, sir. When compared to neem oil cake, the Mahu oil cake have the NPK ratio is very high, sir.
Okay.
Anybody else? Okay, thank you, Mr. Ayyan Durai. Now I request Jaypala. Question? கேளுங்கப்பா அப்புறம் தான் முதல்ல கேட்குறான் அப்புறம் சொல்ல உட்காந்து கேளுங்க
Ayandre, is it audible?
Sir, is it audible, sir?
Yeah. What kind of extract have you used for GCMS analysis?
Sir,
Whether it is a
hexane.
chloroform?
Sir,
What?
hexane, sir. Hexane, sir.
Hexane extract. Okay. And if you refer some articles, this hexadecanoic acid, octadecanoic acid, this will be a major attractant for many pest insects.
Sir, many pest. I can't
Yeah.
hear. Sir, I get not clearly. Pest and
Yeah, these hexadecanoic acids and octadecanoic acids will attract insects. So, if we are using this extract for suppressing fungus, then it will favor insects. So, what is your opinion?
Indra, do you able to follow what he is asking?
Yes,
This
yes, sir, yes,
octadecanoic acid will attract the insects. If you use Magua cake for the management of pathogen, the
Yes, sir.
insects will be attracted.
Yes, sir. Yes, sir.
So unknowingly or knowingly, you are just introducing the problem of pest.
Yes, sir. But yes, sir.
That's what he is asking.
Yes, sir. But I am not observed any that pest and this pest population in that particular my pot culture and other experiments, sir. But I know, sir. But these compounds are act as a attract by the pest. But I not noticed any pest in that my pot culture experiments, sir.
Okay, right. Thank you. Now I request the next speaker, Jayapal. Jayapal.
Yes, sir. Available, sir.
Go ahead.
Good afternoon to all present here. I'm here to present my AGSC conference paper entitled Olfactory Response of Legume Pod Borer Maruca vitrata moths to volatiles from different plant pods of Dolichos bean. We all know food legumes forms the important source of protein in our daily diet globally. Maruca vitrata is one among an important pest of legume crops, which are reported to have a most important yield constraint when compared to major pulses and legume vegetables. The readily available management strategies like chemical pesticides is difficult to this day. Since this behavior of the pest is quite difficult, since it's webbing nature and the development of resistance to the most of the pesticides is getting today. So the alternate options like the use of volatile organic compounds emitted from the host plants will help the insect to orient towards the host plant, either for identification, feeding, and for oviposition. Hence, the base of studying the behavioral studies of the insect and the host plant infochemicals, the volatile organic compounds, and the electrophysiological studies by using the electroantennogram paves the way for identifying the source of the volatile compounds which are responsible for attracting the insect is possible. Hence, the methodology includes first the preference of adult moths is tested by using the Y-tube olfactometer for both males and females. Here, I have used 30 individual number of adults with 11 different combinations. The combinations includes between the healthy flower buds and the infested flower buds, healthy pods and infested pods, and both the healthy pods and flowers, and also the whole plant also. The experimental setup goes like this, like the air is generated by using an aquarium pump, which passes through the humidifier, and the air is controlled by means of air pressure controller, where 1 to 2 ml per liter of air is passed through the water source. where the odor compound is present, which passes through the white tube. At the end of the white tube, will release the adult moths and test it for the preference. And also for the whole plant preference, I use the setup. And the second part of the work is like the collecting the volatiles from the different order different plant parts by using the air entering method. The absorbent material for collecting the volatile organic compounds is Sporopac Q, and the compounds are eluted with the solvent diethyl ether. The different flower parts used were the open flower buds, unopened flower buds, 20 days old plant, and the whole plant. The collected volatile compounds were injected in the GC coupled EAG, where the injected sample is split to the detector and also the EAG. Here we have used the live the excised antenna from the adults were dissected and carefully placed in the probe of EAG and tested. The results include regarding the olfactory response with males, the females, the female moth shows higher preference towards the flower buds when compared to the other parts like pods and the whole plant. Since this is mainly due to the ovipositional site, most of the mostly the Maruca vitrata prefers the flower buds for oviposition. This might this is the reason for the preference. And when it comes to the male moths response, the males shows more response to the healthy plant and also to the flower buds. This is mainly due to the males makes this as the orienting towards the host plant. Then comes the results of GC EAG. Among the different volatiles emitted from the headspace, here I have shown the results of headspace volatile from the flower buds, which shows the most preference. Hence, among the 14 different compounds, osmin, linalool oxide, linalool, decanol, N-decanol, beta-carotene, and alpha farnescens has more EAG response. EAG response to the volatiles. So this could be combined with the pheromone compounds. The pheromone, I have also collected the collected the pheromone from the Maruca vitrata female moths by using crude method, where this EE1012 hexadecadienol is a major sex pheromone, and the second one is the hexadecadienol is the minor compound. These two are the sex pheromone of Maruca vitrata. By combining the EAG responsive volatile organic compound with the sex pheromone, we could able to attract more females and also males. Then, with respect to the conclusion, the Maruca vitrata females prefers the healthy unopened flower buds. Further, the responsive flower volatile compounds in the GC-EAG can be blended with the sex pheromone for the better management. This will reduce the usage of insecticides, and also in future the trapping of both male and female is possible, because the sex pheromone will attract the males, and also by using the volatile organic the plant volatiles, the males get the females get attracted. Hence, we are attracting both the sets of pests. This will improve the efficiency in trapping of the pest. And also there are reports shows that the volatile organic compounds which attracts the Maruca, which will also have a preference to other lepidopteran pests like helicoerpenol. So by using incorporating the volatiles with the sex pheromone, we could able to attract some other group of Lepidopterans also. This is also made possible. Thank you.
Okay, now it's open for discussion.
Jayabal, this is a very good presentation.
Thank you,
So
sir.
my compliments to you for very clearly presenting this one. And my appreciation is on the content and the experimental plan setup. Everything is perfectly okay. And
Okay,
coming to the finding part, you have
Yes, sir.
identified two compounds. One is a major compound, and other is a minor compound. The major
Yes, sir.
compound is commonly seen in all the cases.
Yes, sir.
This is a generic volatile organic compound that we come across very frequently when you
Yes, sir.
scan the library.
Yes,
And
sir.
how come this is so significant in attracting Maruca vitrata? Because Maruca vitrata in the last decade or so, it has assumed the status of a major pest.
Yes, sir.
And now you are pointing out that this major compound alone is capable of attracting this one. Could there be any other compounds that synergize this one?
Yes, sir. Sir,
What are the other compounds?
Sir, actually, reports are there that like 1-octane triol has a synergetic effect with the pheromone, sex pheromone, sir. But with respect to Dolichos vein, when considered, that compound was not was present mostly, sir. And also the females has more response towards these compounds, sir. This differs with plant to plant and the host, sir.
Okay, if that be
Yes,
the
sir.
case, it is only a blend that is going to help you, not a
Yes, sir.
singular compound that will be sufficient enough to
Yes, sir.
cross an attraction. Okay, and
Yes,
also
sir.
when you use this compound
Yes, sir.
along with other lepidopteran pheromones, then
Yes,
there's a
sir.
possibility that the contamination is there. Okay, particularly
Yes,
when you
sir.
prepare the pheromone blend,
Yes, sir.
the isomer ratio is very, very important.
Yes, sir. Yes, sir. Yes,
And
sir.
by adding an extraneous compound like this,
Yes,
for
sir.
example, you are citing the example of Helicoverpa armigera.
Yes,
What will
sir.
happen is that there's going to be a case of a contamination. That is number one.
Okay, sir.
And number
Yes, sir.
two is that there could be a possibility of failure in case of registration, because these things have to undergo registration. Therefore,
Yes, sir.
that may not be
Okay,
a
sir.
correct approach. But
Yes, sir.
in behavioral control, we always
sir.
say that you can mix them at the field
Yes,
level,
sir.
like a tank mix formulation, and
Yes,
that
sir.
could be the approach that we have to look forward
Yes,
in
sir.
this one. What about the linalool here?
Sir, this is also very effective, sir. Linalool also has a higher response, sir. In the case, the females only shows the higher response, sir, when compared to males. The females shows a higher response, sir. Here,
Okay.
this graph is the result of the response of females, sir.
See, generally in the case of the parapheromones,
Yes, sir.
linalool plays a major role. Okay.
Okay, sir.
And
Yes, sir.
it is only the ratio of the blend that is going to decide. Do you
Yes,
have
sir.
any
Yes, sir.
plans to work on this blend in your thesis program?
Yes, sir. Yes, sir. That I have planned, sir. I have to test this compounds individually with different ratios, and also when combining with the sex pheromone also with the different ratios, then only we can come to a conclusion which concentration is effective, sir. Both in bioassay at the lab and also in field, sir.
Okay, that's good. And
Yes,
you have absolute clarity over the subject. Very good.
yes, sir. Yes, sir. Thank you, sir.
Any other question? Any other question from the participants? So if not, thank you.
Thank you, sir.
Now I request Lekha Priyanka.
A very pleasant good afternoon to respected all. Welcome to my presentation on the topic Parasitic Potential of Telenomous Remus on Sporadoproliterics in maize ecosystem. The one which is displayed on the left side is the egg mass of fall armyworm, that is Sporadoproliterics, and the one on the right is Telenomous Remus female parasitoid adult. So these two are the key components in which all the experiments revolve. So let us see one by one. First is, I would like to explain the sexual dimorphism in Telenomous remus. The reason for explaining this is, this forms the base for all the experiments. So the sexual dimorphism observed in Telenomous remus is that in female, we can find the ovipositor very clearly, whereas in males, it is absent, which we all know, followed by which the shape of the antenna has to be noted. That is, the scape or the basal antennal segment in males is longer or lengthier, whereas in females it is shorter. Next is the type of antenna in males is geniculate, and the subsequent antennal segments in males are uniform in size, whereas in females we can find that the antennal segments are enlarged and club-shaped towards the tip, and the antenna has serrations too. So these sexual dimorphic characters are very well visible under the stereo microscope, based on which mated female adults were taken and the experiment was carried out. So the first experiment aimed to ascertain the parasitoid host ratio, which is the key step for any successful biological control program. We tried different parasitoid host ratios, namely 1:10, 1:20, 1:30, 1:40, and 1:50, out of which the ratio 1:40 proved to be very effective. Here, percent parasitization as well as percent parasitoid adult emergence was found to be maximum. So, there are several reasons behind this, which I would like to explain as a justification for my result. First is, at the ratio 1:50, there was emergence of few larvae, which indicates that the parasitoid is not able to parasitize those many number of eggs, followed by the ratios 1:10, 1:20, and 1:30. Though there is parasitization, maximum parasitization compared to 1:40, parasitization is nothing but blackening of eggs was observed, and percent emergences, the number of adult parasitoids emerging from the eggs was observed. So, though in the first three ratios there is maximum parasitization, there was no emergence of adults. The reason is superparasitism. Let me explain. First is, the very behavior of Telenomous remus is that it is a cilionnid parasitoid, and it is a potential egg parasitoid of Sporadoproliterics as well. It is a solitary parasitoid, and the mode of parasitism exhibited by Telenomous remus is simple parasitism. That is, it lays only one egg per host, which is making it a successful biocontrol agent as well. Next is, local mate competition is observed in Telenomous remus. That is, the males emerge from the parasitized eggs first, followed by which the females would emerge, and the females will readily mate with the males. So once the female has completed her mating, the female becomes unattractive to the males. So it is more or less that mating occurs only once. Having these ideas, let us look into superparasitism. There are three reasons. First reason is, when there is scarcity of host, superparasitism occurs, which we all know. Followed by that, the parasitoid which we used for the experiment was 48 hours old, which means one day is assumed as the mating period, that is the time taken for mating, and on the next day the female has successfully mated and it was allowed to parasitize. So when it is 48 hours old, the female has readily mated and her abdomen is filled with eggs. So when she is exhausting her eggs through parasitization, and since there are umpteen number of eggs in her abdomen, there are chances of superparasitization. And the third key reason governing superparasitism is that, the parasitoid since it is a solitary parasitoid and spore effect is well developed in it, though when it goes in search of unparasitized eggs or healthy eggs, occasionally when it happens to cross over the already parasitized eggs, the parasitoid occasionally superparasitizes. So these three are the reason for superparasitism, and this has led to reduced adult emergence in the first three ratios, whereas in the fourth ratio there was maximum adult emergence, and fifth one was less since there was emergence of larva. This was the experimental setup. The egmas of fall armyworm was glued to paper strips, and 48 hours old mated females were allowed to parasitize the eggs, and data was recorded. And this is the video showing the emergence of Telenomous remus. Next is similar experiment was conducted in pot culture condition, again to ascertain the ratio. Here the ratio 1:20 proved to be effective compared to the other ratios. Here percent parasitization as well as adult emergence was found to be maximum. The experimental setup was that 15 days old maize seedling was provided for oviposition substrate to mated female moths of fall armyworm, and the moths were allowed to lay eggs, followed by which the pot was covered with mylar film cages and parasitoids were released and allowed to parasitize. After collection of eggs, percent parasitization and adult for a period of 24 hours, and after 24 hours the parasitized eggs were collected and further data were recorded. I will show the experimental setup shortly. Next is the laboratory experiment. We conducted a laboratory experiment to test the efficacy of Sporadoproliterics on efficacy of Telenomous remus in parasitizing Sporadoproliterics. The purpose of conducting this experiment is that since Telenomous is proving to be a very effective parasitoid of fall armyworm, we want to mass multiply Telenomous remus by various means. So we just tried whether Sporadoproliterics can be deployed as an alternate host or not. So since the ratio 1:40 proved to be effective in the laboratory experiment, that ratio was taken, and eggs of both fall armyworm and Sporadoproliterics were subjected to parasitization by Telenomous remus again, 48 hours old mated female, and the result revealed that there was 93% parasitization of Litura eggs, which was next in rank to Spodoptera frugiperda eggs. But anyway, both were almost on par with each other. The successfulness of any biocontrol agent is in that how far its progeny is capable enough or fit enough to parasitize the host again. So to test the host parasitoid interaction and to test the parasitoid fitness of the progenies that are emerging from Sporadoproliterics, the second experiment was conducted. That is, the progenies that were emerging from both Sporadoproliterics and Spodoptera frugiperda eggs were taken separately. And again, these two progenies were provided with fall armyworm eggs. The purpose was that the progeny which is emerging from Sporadoproliterics, whether the progeny that is emerging from Sporadoproliterics is capable enough in parasitizing the fall armyworm eggs was to be assessed. That was the purpose. So to test the host parasitoid interaction as well as the progeny fitness of the parasitoid, as well as the parasitoid fitness of the progeny, these two were again provided with fall armyworm eggs, which again revealed that the progeny emerging from Sporadoproliterics eggs were next in rank in parasitizing fall armyworm eggs compared to the progeny emerging from fall armyworm eggs. But anyway, again both were on par with each other. So with this experiment, I would like to conclude that Sporadoproliterics can be very well deployed as an alternate host for mass culturing Telenomous remus, because the number of female parasitoids emerging from Sporadoproliterics and also the developing duration is similar as that of fall armyworm eggs as well. So this can be effectively deployed as an alternate host for mass culturing. Also, the successfulness of this experiment was tested again, and currently I am maintaining mass culture of Telenomous remus. We are observing about more than 98% parasitization at the ratio of 1:40 in both fall armyworm eggs and Litura eggs. Next is parasitic potential of Telenomous remus on egmas, which were on egmas of fall armyworm, which were laid on maize seedlings of different ages were tested. The ratio adapted was 1:20 because in the previous experiment, yes. In the previous experiment, the ratio 1:20 proved to be effective. So 15 days old crop and 25 days old crop exhibited maximum parasitic potential. Here the result is higher the younger the plant, higher the parasitic potential. The reasons are the morphological and biochemical traits of the plant as the plant grows older influences the attractancy of pests as well as the parasitoids. And the purpose of conducting this experiment was at which stage of the crop the parasitoid has to be released has to be assessed. So this experiment was conducted. This was the experimental setup, and the last and final experiment is that the pesticides recommended for fall armyworm were tested to assess how far it is safe to Telenomous remus. Here, the egg masses, moths were allowed to lay eggs in 15 days old maize seedlings, and over the eggs, pesticides were sprayed. After that, parasitoids were allowed to parasitize. The egg mass sprayed with chlorantraniliprole exhibited maximum parasitization and adult emergence, which was almost on par with control, which reveals that chlorantraniliprole is safer, followed by azadirachtin, emamectin benzoate, flubendomide, and finally spinetoram. So, through my experiments, I would like to conclude that Telenomous remus can be effectively deployed as a biocontrol agent of fall armyworm, which is cost effective as well as eco-friendly. And further field validation works are under progress, with which I will be able to report the field parasitic potential also very shortly. Thank you for the opportunity.
Okay, now the floor is open for discussion. My question is, the ICAR, in their recommendation, they are recommending Trichogramma pretiosum.
Yes, sir.
Have you compared the efficacy of Trichogramma pretiosum with this Telenomous remus?
Yes, sir, I have done, sir. In pretiosum, there is blackening of eggs, sir, but there is no emergence of larva or the parasitoid. Only blackening of eggs is observed, sir. Further, I'm trying in combinations again, sir, to test the conformity.
When it has to be released in the field, Telenomous?
Sir, during evening hours, egg laying.
No, not in the period of the crop growth. I'm just
15
asking.
and 25, when the crop is younger, sir. 15 and 25 days
days
old. That is up to 35 days old, it is proving to be effective.
In fact, in 25 days old maize, we are recommending spinotirum.
Yes, sir.
But in your studies, spinotirum is
not safe.
Not safe. In
yes.
that case, what we have to do? The first, second window,
second window, late phase.
Second, third phase.
Yes, sir.
What is our recommendation in the first window?
Right now, in the first window, we are taken up chlorantraniliprole. The second window, either chlorantraniliprole or flubendomide.
What is the effect of flubendomide here?
Sir, flubendomide is hazardous only, sir, but chlorantraniliprole is very safe.
It is highly safe.
That is what we use frequently, sir.
But now, in that case, we have to say that if we use this Telenomous remus, then we have to give only the chlorantraniliprole.
We have given chlorantraniliprole, and if you see the residual toxicity of flubendomide and chlorantraniliprole, by nature is safe. Flubendomide also, the residual toxicity gets lower and lower. That is why in the second window, in the first phase, early stage of this one, we are recommending. So we are capitalizing on this. This is how we are progressing.
About the multiplication of
did
this,
you test pretiosum or mavanzai?
Sir, it is in progress, sir.
Which one you are testing? Earlier in your presentation, we have seen that pretiosum doesn't parasitize.
Yes, sir.
But now you are saying that pretiosum is able to parasitize. Is it so?
No, sir. During that time also, I said that there was no emergence of either parasitoid or larva, but there is discoloration of the egg, sir. And when I dissect the egg and see, I'm able to see underdeveloped pretiosum. Emergence is a problem, sir. I will try with different temperatures, and soon I will produce the report the results.
In which layer you have taken the egg? From which layer? 3 or 4 layers of eggs, fall armyworm eggs will be there,
Yes, sir.
From which layer you have taken?
Sir, pretiosum I tested single layer, 2 layers, and regular fall armyworm egg mass. But for Telenomous, The EGMAS as such it was given, sir.
Yeah, because it is able to penetrate very deeply.
Mm.
We can use it. And when we started the work with the Telenomous Remus, the parasitism was dismally below 20%. And now we are able to reach somewhere around 95 or 98%. And now you are suggesting
Yes.
Sporadoproliterics as the
Alternate host.
is it a factious host or alternate host?
It is actually a factious host only, sir.
Now, what is the advantage of the factious host?
Cannibalism is almost less or nil, sir. Next is easy to mass multiply. Since in Frugipeda we are observing cannibalism and easy to mass multiply. We have several hosts for Litura. And the EGMAS is also similar in appearance just as fall armyworm makes. So it is able to parasitize very effectively. And one more key advantage in Litura is that, sir, in fall armyworm makes, we are getting emergence of Telenomous Remus for only 4 days. Up to within by the 4th day, emergence is getting stopped, sir. But in Telenomous, up to 6 days, we are getting emergence of parasite.
Is it advantage or disadvantage?
Sir, advantage, sir, because when there is high emergence and for many days, if we are getting emergence, I suppose it is advantage, sir.
It's definitely an advantage.
Yes, sir.
Because if you see the broods emergence in the case of fall armyworm, you get a continuous broods.
Is
it
yes,
not?
Sir. Yes, sir.
Which means that these overlapping generations will be there. And here there is a continuous emergence of parasite. That means that you are going to get a prolonged
period
control.
of parasitism.
No?
Yes, sir.
Very good. That's it.
Any questions from the audience? Anybody? Okay, thank you, Dr. Mr. Lekha Priyanka, for your nice presentation. Now I request Dharani Priya.
Thank you, sir.
Good afternoon, sir. I'm here to present my research findings on synergistic toxicity of marine macroalgal extracts with azadirachtin and beta-asterone mixtures against cosmopolitan pest Sporadoproliterics.
Can you please explain about your
Sir, myself, Dharani Priya, and Dr. Raghuraman sir is chairman, and Dr. Bhuvaneshwari ma'am and Dr. A. Lakshmanan sir and Dr. K. Chandrakumar sir are members. Okay. As a universal nothing, we all know the indiscriminate use of synthetic pesticides causes hazardous to the environment and also various ill effects. So to overcome this, we are switching over to the use of biorationals that includes botanicals. Interestingly, the plant extracted compounds shows various mode of action, and also it proved to be it has various biological activities. Our focus is to use the marine macroalgal extract, marine algae, that is outstanding group of marine organism in pest management. The marine algae contains various secondary metabolites that has a insecticidal action that can be used in pest management. And azadirachtin, the active ingredient from Azadirachta indica and beta-acerone from Acorus calamus, it also possesses various biological activities. And when it is combined with macroalgal extract, it possesses enhanced toxicity. So with this background, our objective is to study the toxicity and the combination toxicity of azadirachtin, beta-acerone, and macroalgal extracts against the test insect Sporadoproliterics under laboratory conditions. And the materials we used is macroalgae. The macroalgae are collected from Hare Island, which is a part of Gulf of Mannar Marine National Park. They are collected during a morning hours where there is a low tide and the water will move away from the shore. At that time we collected. They are collected by hand picking method and washed. They are shade dried and stored. And there are 3 groups of algae are present: red algae, brown algae, and green algae. Red algae and brown algae shows most bioactive compounds compared to the green algae. So we selected red and brown algae. And for screening, we selected 3 species from red and 3 species from brown algae. From red algae, Amphiroba fragilisima, Acanthopora spicifera, Gracilaria corticata are selected. And in case of brown algae, Padina pavonica, Turbinaria conoides, and Sargassum waiti are selected for screening. And after screening, the Sargassum waiti from brown algae and Amphiroba fragilisima from red algae shows a superior insecticidal action compared to others. So we selected that, and it is combined with azadirachtin of 12% purity and beta-asterone of 75% purity for enhanced toxicity studies. So the treatment used here are azadirachtin alone and beta-asterone alone, macroalgal extracts alone, and azadirachtin and beta-asterone is combined, and azadirachtin and macroalgal extracts are combined, and beta-asterone is combined with the macroalgal extracts, and the combination of azadirachtin, beta-asterone, and macroalgal extracts. The macroalgal extracts are prepared by using ultrasonication technique, and the samples are prepared, and it is sprayed in a castor leaf disc of known diameter, and it is fed to the 4 hours pre-star second instar larva of Sporadoproliterics, and data on mortality was recorded every 12 hours up to adult emergence. And it's a graphical representation of each treatment. When azadirachtin alone is used, there is the 50% mortality was occurred from 500 ppm, and the maximum insect growth regulatory action was observed up to 800 ppm. Whereas in beta-asterone, the maximum, the 50% mortality was occurred between 1700 ppm. And macroalgal extracts, Amphiroba fragilisima and Sargassum waiti shows superior insecticidal action. Similarly, in azadirachtin and beta-asterone, when it is combined, 800 ppm and 1950 ppm shows a maximum mortality, maximum insect growth regulatory activity. And azadirachtin is combined with seaweeds, Amphiroba fragilisima and Sargassum waiti. It's at 800 ppm, it shows a maximum mortality. Whereas when we combine azadirachtin, beta-asterone, and macroalgal extracts, it shows a 70% and 90% of mortality in case of Amphiroba fragilisima and Sargassum waiti, respectively. This table clearly shows how it enhanced toxicity. When azadirachtin alone is used, the LC50 is 549 ppm. But when it is combined with seaweed extracts, the LC50 value, the toxicity was increased. Similarly, beta-asterone, when it is used separately, it is the lethal dose was 1740 ppm. But when it is combined with the seaweeds, it shows increased toxicity. Likewise, when seaweeds are both seaweeds are used separately without addition of azadirachtin and beta-asterone, the LC50 value was more. And azadirachtin, beta-asterone, and Amphiroba and Sargassum, when it is when 3 are combined, the Sargassum waiti, the LC50 value is 211. So this clearly explains it. It has the macroalgal extracts, when it is combined with azadirachtin and beta-asterone, has enhanced toxicity, shows enhanced toxicity. And this is pictorial representation. The azadirachtin shows IGR action and the dead larvae. So finally, I conclude that the macroalgal extracts, when it is combined with azadirachtin and beta-acerone, it shows an enhanced toxicity, and it can be included in the integrated pest management studies. Thank you.
Now it is open for discussion.
Dharni Priya,
Sir.
You get back to the second slide. That is your treatment schedule. Methodology, treatments, azadirachtin alone.
Yes, sir.
Castor leaf disc, azadirachtin alone. How did you fix the dose?
Sir, we have fixed from 1000 ppm, 100 ppm to 1000 ppm, sir.
On what basis?
On the, sir, from previous literatures.
No. Did you conduct any study ranging assay in the laboratory?
So that is the ranging from 100 to 1000. We first. It's for screening. We selected that.
Okay. What about the slope value? Then R value for your LC50?
So I have
for
not
individually. See, you are interested to know what is the combination effect. How do you express the combination effect? It can be, if it is less than 1, what is it? If it is more than 1, what is it? It can be a direct antagonism or a direct synergism. If it is a case of a synergism, it can be a subadditive synergism or potentiating synergism. Nothing has come out from your findings. When you test a compound, 2 compounds means LC25 plus LC25. The summation should give the value. If you get a mortality more than LC50, then you can say that there is a case of a synergism. There you have to categorize subadditive, temporal, spatial, like that. Okay. When you are going to take up 3 compounds, then LC10, LC10, LC10. This is how you have to progress. And I think the structure has to be looked into very carefully. And what is the premise for combining? Normally, when you combine products, binary combinations are being used. Here you have gone for 3 different compounds, which means that azadirachtin by itself, it has so much of value. Beta-acerone, then macroalgal extract, it's an unclarified material. What is the purpose of combining all these things? So many compounds. And is it, will it have any field applicability? This is my question to you.
We are doing research for taking the results to the field, not for academic purpose. Whatever finding we are getting it from your thesis, a PhD work, that has to be taken to the field. Okay? So in that case, you have to see whether these things are available in the market or not. When we say that you have to take up the spraying of these things, you have to assess the availability also.
Okay.
Any more thing?
Hey, okay.
Yeah.
Priya, you mentioned that red algae and the brown algae contains more bioactive compounds than green algae. Why? Why red and brown algae contains more bioactive compounds?
Sir,
what is the reason?
Sir, green algae, the photosynthesis activity was more compared to this red and brown algae, sir. And red and brown algae, based on the tannin content, is also higher in red and brown algae compared to green algae, sir. For that reasons, maybe the. No, sir, that is the reason, sir.
Priya, your MSc work was also on algal forms. Now you are the master of this subject. You have to tell us what are the compounds.
Thank you.
Thank you. Any more questions? Okay, thank you. Now I request Praveen. Praveen,
Yes, sir. Yes, sir.
I hope that you have presented the same thing at Ichangote also. Is it not?
Sir, no, sir, that is different, sir.
No.
Something.
What difference it is there? The same thing I could
Yes,
able
sir.
to see.
Yes, sir. Sir,
Then
there
now
I
you are
have,
saying that it is a different one.
sir,
How can you
that,
present the same thing in two different places?
sir,
Thinking
no, sir.
that I may not be the chairman for this particular session, is it not?
Sir, that is, I have present only alone the Mintas Picata alone, sir. Here I'm going to Mintas Picata alone, I have shown there, sir.
Here, what are you going to present? The same, I think the same title is being
more or less
used at Ichangote also.
No, sir. Sir, there I have present that antifungal activity, sir, for Mintas Picata alone, sir.
So here, what are you going to do?
Sir, here I'm going to present about begin the mechanism of action.
See, the
So,
title remains the same.
The same title is being used, I think, is it not?
Sir?
For Ichangote, what is the title you have presented?
Sir, antifungal activity of Mintas Picata volatilum against
here you have put the volatilomes mediated. You have added only two words.
Sir.
Okay, let us see.
Shall I start presentation, sir?
Go ahead.
Sir?
Present பண்ணுங்க.
Yes, sir. Yes, sir. Is my slide is visible, sir?
It is visible.
Yes, sir.
Make it full screen.
Yes, sir. I have done, sir.
Okay.
With respect to Dean SPGS, Director CPPS, and Professor and Head Department of Plant Pathology and Agriculture and Entomology, Professor, Associate Professor, and Assistant Professor, and friends. Good afternoon to all. Now I am going to present my research paper on volatilome-mediated antifungal activity against Fusarium wilt infecting tomato. Coming to the introduction, the volatilomes are a group of volatile and organic and inorganic compounds produced by many organisms and also produced from the natural ecosystem. So these volatile organic compounds are low molecular weight, carbon structured organic compounds which diffuse in their atmosphere under high vapor pressure. In fact, these volatile organic compounds are normally produced by the leaves and roots of plants and also in soil and also by beneficial and harmful microorganisms. But their communication between each other against the plant pathogen becomes unknown. But these volatile organic compounds could have different biological and ecological functions in inducing the defense mechanism against the plant pathogen, which also promotes the growth in plants. In order to identify the nature of volatile organic compounds in pathogen suppression, the objective was to find out the important antifungal volatile organic compounds against the Fusarium wilt infecting tomato crop. In our experimental findings, the leaves of volatile producing medicinal herbs were collected and tested for their antifungal activities against the mycelial growth of Fusarium oxysporum using sealed plate method. The volatilome produced by the leaves of Mintas plicata inhibited the mycelial growth of pathogen to an extent of 45%, followed by the volatilome produced by the Symphophagous citratus and Vitis negundo. Based on this preliminary screening, the volatilome produced by the leaves of Mintas plicata, Symphophagous citratus, and Vitis negundo were trapped and analyzed using air entrainment technique. The experiment was performed for 5 to 6 days to trap the volatilome emitted by the leaves of plant samples. Then the trapped volatilomes were gently eluted by adding 200 microliters of HPLC grade ethanol up to 1 microliter, and they were collected in a GC vial and they were subjected to head-phase Gcms analysis. From the result of head-phase Gcms analysis, the volatile compound produced by the effective samples were comparatively analyzed using the heat map. So in this heat map, The compound carbon was found to be the most abundant volatile organic compound produced by the leaves of Mentha spicata, which is having high relative abundance. Similarly, the geraniol and citronola were also produced by the leaves of Symphophagous citratus, which is also having high relative abundance. Based on the efficacy of selective volatile organic compounds obtained from the medicinal herbs, a standard compounds such as carbon and citronola were tested individually for their possible inhibitory effect on mycelial growth of pathogen using bipartite plate assay. The volatiles emitted by standard compounds were compared with volatile extract of plant samples. In this bipartite assay, the carbon completely suppressed the mycelial growth of pathogen at a concentration of 500 ppm, followed by the volatiles of citronanol. Similarly, the volatile extract of Mentha spicata did not allow the pathogen to grow at 500 ppm, and it is also followed by 98% incubation at 400 ppm, which causes this abnormalities in hyphal growth and look flocose appearance with no spore germination. This is compared with to the normal growth of mycelia in unexposed control plate. So this experiment was followed by the volatilum extract of Symphophagous citratus, which also inhibited the mycelial growth up to 90% at 500 ppm. In order to identify the mechanism of action behind this, the mycelia of Fusarium osteosporum were completely distorted and reduced in size than citronolol and Symphophagous citratus. The sporulation was also significantly altered when exposed to the volatiles of Mentha spicata compared to control. Interestingly, the emergence of germ tube from Macroconidia was also arrested with extreme variability in size and shape when exposed at 500 ppm concentration. In this, the vapor phase mediated assay are also carried out to characterize the behavior of post germination of pathogen and also to assess their antifungal activity using 96 well microtiter plate. Here, the 200 microliters of pore suspension was added to all the wells except the center 4 wells as volatility center. This analysis was observed periodically up to 150 minutes. The spores of Fusarium osteosporum were significantly incubated away from the volatility center throughout the microtiter plate, which indicates that vapor phase of Mentha spicata diffuses in the entire plate even up to 150 minutes of observation. Therefore, the volatile extract of Mentha spicata alone exposed to plate were found to be the fungicidal action against the spore germination of Fusarium osteosporum. So in order to develop a suitable volatile formulation, the leaves of Mentha spicata were immobilized in a vermiculite ball mixed with a castor oil as a volatile immobilized vermiculite ball formulation. Here, the vermiculite was served as a volatile absorbent material. Under the same analysis of vermiculite ball formulation, the expansion of crystal structure was observed from the volatilums of Mentha spicata immobilized ball. Under this observation, it significantly increased the absorption efficiency with vermiculite content. This expansion is mainly due to changes in the structure of octahedral and tetrahedral layer in the Mentha spicata immobilized vermiculite ball. Hence, the volatilums of Mentha spicata leaves immobilized vermiculite ball was studied further to find out their efficacy against pathogen. The efficacy of Mentha spicata immobilized vermiculite balls was tested against Fusarium osteosporum on an olfactory sample. The vermiculite ball was kept at the center of the olfactory sample. The pathogen was fitted in all outlets. The volatiles of Mentha spicata flow equally to all the 6 outlets and incubated up to 92% of mycelial growth of the pathogen. These volatilums not reduced the size and shape of the conidia, thereby arresting the growth of germinating hyphae. This was compared to control as shown thick and fast mycelial growth with fluffy appearance in an unexposed control plate. From this research finding, it was concluded that volatilum produced by the leaves of Mentha spicata immobilized vermiculite bud effectively reduced the mycelial growth and also the correlation behavior of Fusarium osteosporum. A pure form of volatile organic compound such as carbon and citronol is found to be less effective than the volatile extract of Mentha spicata. Based on these findings, the cultivation of Mentha spicata as intercrop with tomato could reduce the spread and severity of Fusarium wilt pathogen and also promote the growth of plants because the mint volatiles will emit and suppress the growth of pathogenic organism under field conditions. This will benefit the farmer as dual crop cultivation of mint and tomato production. In future, it is necessary to optimize the volatile organic compound trapping and in need of robust technology for non-targeted profiling under field conditions. Apart from this, a different mode of delivering method is also required, such as direct application to soil, root immersion, and seed priming and aerial spray. Further, the defense pathway in activation of salicylic acid and the jasminic acid upon volatile formulation need to be analyzed. Hence, the Minthas induced by phytobiomolecules could be explored for the suppression of plant diseases, as earlier reported for fungal biomolecules such as strobilurin from the mushroom Strobilurus tenacellus. Finally, I acknowledge the Department of Plant Pathology and Department of Agriculture and Entomology TNAU for supporting during my research work. Once again, I thank you for giving us such opportunity to present my research work in this conference. Thank you. Thank you, sir.
Okay, now the floor is open for discussion. Any questions from participants? Okay, if not, thank you, Praveen.
Yes, sir. Thank you, sir.
So on behalf of the entire AGSC team, we would like to thank our beloved professors who spend their valuable time for the successful conduct of this particular plant protection theme in a successful way. So we thank all of you, sir, for your valuable presence here. And we thank all the both online and offline participants who have come over here and joined in the successful conduct of this theme number 3 of plant protection. So those who want to present on this poster presentation, please come and join at the specific link which has been provided in your email, and come and join at the time of 5 p.m. today. So all you can leave now. Thank you.